Hello, this is Rachel Hughes, and this is Crash Course Biology. Today we will study cells.
Silly computer, not that kind of cell. That's it, that's the one.
Believe it or not, all living things are made up of millions upon billions upon trillions of little cells that are constantly replacing each other. So when you think about it, we're all just really one shifting block of little tiny specialized structures called organelles, and this jelly-like stuff in between them called cytosol that never stays the same for 2 days together. But we'll get to all that. First, let's start at the beginning.
If you first thought of a prison cell when they first said the word cell, you actually weren't too far off. In 1665, when Robert Hooke coined the name cell, he was observing cork cells, and he named them a cell because they reminded him of the cells or rooms in a monastery.
Now, the cork cells that Robert Hooke was looking at were a type of plant cell, but there are many other types of cells as well. For example, plant cells, animal cells, and fungi and protists, they are all considered eukaryotic cells, which means that they have a membrane-bound nucleus and other organelles besides ribosomes. There are also what we call prokaryotic cells, such as bacteria, which have a nucleoid instead of a membrane-bound nucleus. The nucleoid is the region in which all their DNA is held. They also only have ribosomes as their organelles. Oh, Miss Hughes! Will we have to know how to differentiate between different prokaryotic cells on the test? No, me from the past. You won't have to do that for the test. They get way too complex with all those different types of bacteria. They're all so unique. However, we will need you to be able to identify different parts of plant and animal cells. So to do that, let's go to the thought bubble.
When you first look at an animal cell under a microscope, you might first see the round outline of a cell. Zoom in a little more and you'll see the nucleus, the most recognizable part of the cell. This stores genetic information and controls metabolic activities and could be likened to the brain of the cell. The other organelles of a cell are much harder to see with a compound light microscope, so unless you have millions and billions of dollars laying around and you're just like, hey, let me get myself the best microscope around, you might just want to stick to the nucleus. Within the nucleus, there's the nucleolus, which makes ribosomal RNA, and the nuclear envelope, that's the membrane that surrounds the entire nucleus. There's also chromatin, which is DNA and protein that will coil to form chromosomes right before cell division. Throughout the nuclear envelope, there are little itty-bitty holes called nuclear pores. These allow RNA out of the nucleus and proteins into the nucleus, so that the nucleus doesn't, starve. The entire area between the plasma membrane and the nucleus is called the cytoplasm. This means that this, and even this little guy over here is all included in the cytoplasm. However, the fluid in between all of the organelles is called cytosol. Now let's go back to our red friend over here. This little guy is called the mitochondria. He breaks down sugars to produce ATP, and this means that he is involved in cellular respiration. There's a good reason that he is called the powerhouse of the cell. The next group of organelles that we will discuss are parts of the endomembrane system. This consists of 4 main parts: the nuclear envelope, which we have already discussed, the endoplasmic reticulum, or the ER, the Golgi apparatus, and the vacuoles. The endoplasmic reticulum, or the ER, and no, that doesn't mean emergency room, is a system of membranous channels and saccules. The ER is divided into 2 parts based on the presence of little organelles called ribosomes. The ribosomes are the location where protein synthesis occurs in a cell. The ER is divided into the smooth ER, where there are no ribosomes, and the rough ER, where there are ribosomes. After proteins are packaged in the smooth ER, they are transported to the Golgi apparatus. The main function of the Golgi apparatus is to package, process, and distribute the vesicles from the smooth ER. Vacuoles are very similar to vesicles in the fact that they both store substances. However, vacuoles are normally much larger than the vesicles. In animal cells, vacuoles can also have little organelles called lysosomes attached to them. These are involved in intercellular digestion and can also be compared to the garbage disposal of the cell. The final large group of organelles that we will be discussing today is the cytoskeleton. This helps maintain the cell's shape and causes the cell and its organelles to move. The flexible protein threads that help give the cell its shape and enable parts or the entire cell to move are the microfilaments. The microtubules, on the other hand, are long protein cylinders that form the rigid skeleton for the cell and also help with moving substances within the cell. The final element of the cytoskeleton is the centrosome, which is the microtubule organizing center. Encasing the entire cell is the plasma membrane. This helps regulate what moves into and out of the cell. Finally, single-celled organisms might need a little extra help in moving around. This is why some of them have developed cilia, which are small, numerous hairs that help in cell movement and encase the entire cell. Other organisms have developed flagella, which is a large single hair that aids in the same purpose.
Thanks, Thought Bubble. Plant cells are very similar to animal cells, except around the plasma membrane there's this rectangular structure called the cell wall, and this gives the plant its rectangular shape. Also, since plants are autotrophs and not heterotrophs, they need to photosynthesize, so they have these little organelles called chloroplasts that help them do so. Chloroplasts are then themselves divided into little tiny structures called thylakoids and grana, but we won't get into all that. Let's just stick to the basics. Another difference between plant and animal cells is that plant cells only have one huge vacuole, while animal cells have a ton of little ones. The vacuole also helps with structure in the plant cell, and it also helps it photosynthesize. And now it's time for the open letter. But first, let's see what the secret compartment has in store for me today. My cell phone? Did you know that cell phones are actually called cell phones because of the way that they receive their signal? The tower in the middle of the area acts like the nucleus and sends out huge signals directing all the calls, just like the nucleus does with all the organelles. Hey look! A message! Dear biology, why did you have to go and name everything names that sound the same? You could have gone from A to Z and had funny names like quarks or xylophones. But no, you had to leave quarks to atoms and xylophones to music. Instead, you had to go with centrosomes and centromeres and centrioles and chromatin and chromosomes and microtubules and microfilaments. You make a girl's head spin. Best wishes, Rachel Hughes. Well, that's all we have for this week, folks. Hopefully you enjoyed learning about what makes up your body. Join us next week for my personal favorite unit, cell division. Mitosis and meiosis, and you get all these chromosomes that split in different phases. It'll be great. Crash Course Biology was made possible by Mrs. Ludwig's Cell Project, written and directed by Rachel Hughes, although most of it was improvised.